Welcome. In the previous class, or in a few of the previous classes, we saw the transfer coefficient approach for the mass transfer of especially oxygen from the gas phase to the liquid phase. And we also looked at how to measure KLA in the bioreactors. That the formulation brought us to a KLA, the volumetric mass transfer coefficient of oxygen in a bioreactor. And we also saw a problem as to how to measure this KLA. Okay. Let us take things further in this class. Supplying oxygen at adequate levels to the bioreactor is always a challenge. Okay. The bacterial or the microorganisms, the aerobic microorganisms, gobble up oxygen. Okay. And to supply at adequate rates is always a challenge. This is because the normal mode of oxygen supply, which is aeration, is inadequate to meet the oxygen need of the cells. Okay, that's the reason here. And augmentation with pure oxygen is usually expensive. You cannot use oxygen cylinders. Oxygen cylinders are expensive. Further, the normal aeration and agitation levels in the bioreactor can result in high shear, right? If you increase the aeration rate, the bubbles are going to come out at much higher rates. They're going to break at much higher rates. That breakage would release a lot of energy, which can shear the cells. So the shear, we all know, are the forces that can tear the cells apart. And therefore, a related challenge is that of providing a gentle enough environment in the bioreactor with minimum shear for the cells to multiply and make products optimally. The liquid phase oxygen supply strategy, LPOS, okay, liquid phase oxygen supply strategy, LPOS, can effectively overcome both the above challenges and is inexpensive. This we have shown. In fact, this is something that we generalized and we have shown. This was published as a paper in 1998. Sriram, Manjula Rao, A.K. Suresh and myself. I was the corresponding author here. Oxygen supply without gas liquid film resistance to Xanthomonas campestris cultivation. This was published in Biotechnology and Bioengineering, which happens to be the most prestigious journal in our field of biological engineering in 1998. The details are given here. Okay. And in this lecture, as well as in probably the next lecture, we are going to see some of our research work in which we have applied the transport principles very effectively. Okay, so the principles can be applied anywhere, in this case to cutting edge research. The liquid phase oxygen supply method provides oxygen using a green chemical hydrogen peroxide, which is added according to need. Okay, that's the way we can go about doing this. The added hydrogen peroxide is converted to oxygen using an enzyme catalase, which is made by the cells themselves. And although hydrogen peroxide is toxic at high concentrations, low concentrations employed in the LPOS do not harm the cells at all. Okay. This is the LPOS method based on hydrogen peroxide to provide oxygen. This is something like this. Okay, where are the papers here? The papers here. Okay, I'll come to the paper in a little bit. Or before I get to the papers, let me tell you this. We have the, I just lost my train of thought here. We have, I was going to tell you the idea, right? See, we have the oxygen or the air bubble from which oxygen gets applied to the liquid, right? That's the case of aeration, the standard way by which oxygen is supplied. So here, if you have all this, you need to go from the gas phase to the liquid phase. In other words, you're overcoming a certain resistance. You can view it that way. You're overcoming a certain resistance for the transfer of oxygen from the gas phase to the liquid phase. And if you look at it closely, that resistance is the biggest resistance for transfer of oxygen in this situation. Okay. So our thinking was, can we get rid of this biggest resistance to oxygen transfer from the gas phase to the liquid phase? If we can get rid of this resistance, then the transfer would be that much simpler, because this was a classic challenge a long time ago. I think it's still a classic challenge. Uh, so the thinking was something like this: as long as there are two phases, as long as there's a separate gas phase and there's a separate liquid phase, there will certainly be a gas liquid film resistance. And if we can get rid of the gas liquid film, then we can overcome this resistance. And the only way to get rid of gas liquid film is not to have two phases, have a single phase alone. Okay, that was the thinking. Or in other words, we were going to generate oxygen in the liquid phase itself as a part of a liquid phase reaction. Thereby, the oxygen species would be in the liquid phase itself, and that would supply oxygen to the cells. Okay. Note this concept of, or this notion of oxygen being a gas. That is fine, it is a gas under normal temperature and pressure, but it is a species, it is a molecule. It can be in the gas phase, it can be in the liquid phase, and so on, depending on the conditions, okay, or even the solid phase, depending on the conditions. In this case, normally when it is present in air, it is present as a gas phase. When it gets into the liquid, it is dissolved oxygen, it is in the liquid phase, and that is what the cells take up. Therefore, the idea was, if we can directly provide it in the liquid phase, then the cells can take up much, or the supply rates can be high enough, and so on. That was the idea that we tried out. Okay. With LPOS, if we write the oxygen balance, it all comes down to the basic oxygen balance now, the balance, mass balance. Input rate minus output rate plus generation rate minus consumption rate equals the accumulation rate. The output rate is 0 because we are looking at broth minus bubbles. The generation rate is there in this case because we are generating hydrogen peroxide, we are generating oxygen using hydrogen peroxide. The input rate is KLa times CO star minus CO2 as given earlier times the volume. Okay, so volume is needed to convert this into moles per time. This is from a hydrogen peroxide generation kinetics. We'll have to put in the appropriate term here. This is the uptake rate in terms of oxygen uptake rate. We need to multiply. This is given usually in units of the form that we need to multiply it by the cell concentration and the volume to get moles per time equals the accumulation rate. Okay. So this is the mass balance, and this equation can be used according to need. When you don't have any cells, this goes to 0. You could use, then this becomes irrelevant. Therefore, these two terms need to be taken together. The catalase that is necessary for generation of oxygen from hydrogen peroxide actually comes from the cells. Therefore, these two terms usually go together. I thought I'll present to you some papers of ours, which initially the first paper would talk about this liquid phase oxygen supply strategy, and then later papers would talk of other things, all related to transport as applied to cutting edge biological engineering aspects. So let's do that. I think this is what I have here, right? Yeah. Let's start doing that. See, this is I think this is big enough for us to look at, isn't it? Okay. So this paper published in Biotech BioEng, oxygen supplied without gas liquid film resistance to Xanthomonas campus cultivation. G. Sriram by Manjula Rao, A.K. Sureshan, myself. I used to spell my name a little differently then, so 1998. It's changed it around 2001, 2002. So after that it is SURAISHKUMAR. Okay, let me read out the abstract to you and then explain a few things here. Okay. Alternative methods of oxygen supply are of crucial importance. Remember, this was way back in 1998, 22 years ago. Alternative methods of oxygen supply are of crucial importance, especially in viscous fermentations and shear sensitive fermentations. A method of oxygen supply that completely eliminates the gas liquid transport resistance has been presented. The method involves a need-based liquid phase decomposition of hydrogen peroxide to provide the necessary oxygen. When Xanthomonas campestris was cultivated, this is a viscous cultivation, using this method of oxygen supply, dissolved oxygen or DO levels were maintained above the set point of 50% throughout the cultivation. Whereas the conventional cultivation was able to meet the culture oxygen demand only for about 6 hours in a 72-hour fermentation. Furthermore, the maximum specific growth rate and the xanthan yields in the novel cultivation were 89% and 169% respectively of those obtained in conventional cultivation. A mathematical model was also developed to simulate and predict results in fermentations employing the presented methodology. In addition, studies with HFCl pre-treatments indicated that monofunctional catalase may be responsible for the decomposition of hydrogen peroxide. These are fundamental details. If you want, you could read those. And we also could back out some constants which are fundamental and so on. Okay, I'll probably read some parts of the introduction to present the base, and then we'll talk about what we did. Okay. The performance of aerobic bioreactors, especially on large scale, may be suboptimal due to inadequate oxygen supply rates. Insufficient oxygen supply could lead to suboptimal productivities as well as products of low quality. All these are well known. Inadequate oxygen supply results due to low solubility of oxygen in the medium, 8 ppm at equilibrium, and the large gas liquid film resistance for oxygen transport when aeration is used. Okay, for people who don't know about the film, if you look at the concentrations of oxygen near the interface, just beyond the interface into the liquid side, assuming that it goes from the gas phase to the liquid phase, just beyond the interface on the liquid side, there's going to be a huge drop in concentration. Okay, then the drop is going to be much, much shallower. So the region where the oxygen concentration drops is the conceptual film, nothing else. Okay, it's a conceptual film. There's actually no physical film there. It is a region where there's a huge drop in the oxygen concentration. Researchers have tried to eliminate the gas liquid oxygen transport to meet the oxygen requirement of the culture through several means. Some of the means are given here. The methods described here have attempted to improve the oxygen transport rates, but still retain the basic limitation of gas liquid oxygen transport. It is always from the gas phase to the liquid phase. Hence, they have been only partially successful or partly successful and may not meet the requirement of large-scale bioreactors, and thus may make the systems partly anaerobic. This could lead to formation of undesirable products in the system, which could question the economic viability of the process itself. Further, if either the fermentation is viscous or the microorganisms are shear sensitive, such methods of enhancing oxygen availability may be ineffective, okay, because the productivity will go down. Therefore, if oxygen transport can be achieved with complete elimination of the gas liquid transport, it would greatly improve the economics of the process. The methodology demonstrated in this article to overcome gas liquid transport resistance is through a liquid phase conversion of hydrogen peroxide to oxygen and water. This reaction is catalyzed by the enzyme catalase available from the culture itself. No external catalase is needed. In this case, the oxygen molecule, being in the liquid phase, is ready for consumption by cells, and hence gas liquid oxygen transport is non-existent. Although hydrogen peroxide is used to kill cells, we all know, 4.5% is used for a mouthwash and so on. The concentrations required for killing cells are in the percent solution range, whereas the methodology presented here employs hydrogen peroxide on a need basis at concentrations at least 150 times lower. Therefore, cell death need not be a concern, and we have also demonstrated that cell death is not a concern. The particular reaction for oxygen evolution was chosen because it occurs naturally. Hydrogen peroxide is generated in the cell either directly by the divalent reduction of oxygen or indirectly by the dismutation of superoxide radicals. The generated hydrogen peroxide is scavenged through catalase. Most aerobes contain catalase, which is used as a defense against hydrogen peroxide generated in the cell. Okay. So we are using something that is already present in the cell to take care of this. Thus far, hydrogen peroxide has not been used as an oxygen source for bacterial cultivation and so on and so forth. And the scope of the paper is given here, followed by some materials and methods, and then some modeling details. Okay. And let's look at some results and discussion here. Let's probably, okay, this is some basic characterization, DO versus time, when we pulsed it with experimental and simulation values for DO variation in Xanthomonas campestris during aeration. Okay, this is the way it varies. We'll not get into that. And then feasibility demonstration of oxygen supply using hydrogen peroxide. Okay, what we did was, let me go to the graph here. We took the medium with cells, standard bioreactor broth, and also did an experiment where we considered a medium without cells. Okay. Our thinking was that somehow the cells would sense hydrogen peroxide, put out catalase, and then break things down. That completely changed, as I'll tell you in the next class. Here, with that idea, we went into this experiment. When we added a 200 micromolar pulse of hydrogen peroxide to the medium with cells, the dissolved oxygen level went up as expected. As soon as we added the pulse, the DO level went up. Okay. This very clearly told us that the oxygen is becoming available in the broth. Okay. We thought oxygen is becoming available immediately in the broth, which the cells can take up. That was different later. Then when we did it with the medium without the cells, of course, there was no availability of oxygen in the broth, no oxygen became available in the broth, no increase in DO took place, or this mild increase in DO is due to surface aeration. Okay. That over a 60-minute period that we can explain through surface aeration alone. Okay, so this was feasibility demonstration for the availability of oxygen from hydrogen peroxide decomposition by cells. This was done. Then we did the cultivation with hydrogen peroxide pulses. Okay. Graduate student sat and did this experiment over a long period of time, and a few graduate students did this. This is prediction, we will not get into that. Here we are looking at dissolved oxygen versus time for a Xanthomonas campestris cultivation. In the conventional aeration case, the conventional method of providing oxygen supply aeration, we started at 100%. By the way, this is percentage air saturation. In air, you have 21% oxygen. So if you reach 21% oxygen in air, you have 100% air saturation. Okay. So in theory, you can go up to 480% air saturation, right? Because if you go to 100% oxygen, it will be almost five-fold, 21. You're talking of 21 and 100. So almost five-fold, 4.89-fold of the air saturation value. And therefore, you can go from 0% DO to 480% DO, because this is percent air saturation. So here we are looking at, if you start out with 100, for a Xanthomonas campestris cultivation done with air, the DO drops to 0 and remains at 0 pretty much throughout. Let's not get into this region. Whereas if you use pulses of hydrogen peroxide, you add a pulse, the DO goes up and then it comes down. When it comes down close to 0, you add another pulse, then it goes up, comes down. Okay, something like this. You add a DO versus time, you add a pulse, it goes up, comes down. When it comes down here, you again add a pulse, it goes up, comes down, and so on and so forth. So by continuously measuring the DO and adding the pulse when the DO is close to 0, kept the DO value above the 50% DO level which we set for this case. Okay, so here we found that in the case of conventional cultivation, the growth rate, which is important because the growth rate will determine the cell concentration, higher the cell concentration, higher the productivity, and therefore this is important. Okay, so the growth rate is comparable between the two. So this method of cultivation is not affecting the growth rate significantly. 0.111, 0.098 R inverse. Then the yield of per unit substrate, the product concentration divided by the substrate concentration, 0.1657, 0.287. It is higher in the case of hydrogen peroxide based cultivation. Yield per unit biomass, P by X, is almost 9-fold. Okay, huge. Each cell is producing 9-fold higher xanthan gum. Okay, and the final xanthan concentration also is much higher than expected. So using this cultivation has significantly improved the xanthan yield from this. We thought it was due to a better supply of oxygen or a better supply rate of oxygen in that situation. Also, this is a viscous fermentation. It is difficult, normally speaking, to supply oxygen to this fermentation. So this method can become very useful in supplying oxygen to both challenging fermentations as well as regular fermentations. We also worked out the costs that are involved, and if you consider everything, the cost per kilogram of oxygen turned out to be 15 paise compared to about 2 rupees per kilogram when you consider aeration as a source. Why? Although air is free, you need to go through appropriate filtration, appropriate sterilization, and so on and so forth. And that is expensive. Okay, and of course the compressor costs are there. There are no compressor costs here. So many other parameters on which we did the comparison. It was 15 paise per kilogram of oxygen. That was 2 rupees per kilogram of oxygen. I think we will stop here for this class. When we come back, let me present a few more applications of transport in our work, okay, so that you can appreciate the width of application and even to cutting edge aspects. Okay. See you in the next class. Bye.